Welcome back to my channel Knowledge Chronicle.
Gene editing is no longer science fiction, it's the future of medicine. But is this revolutionary technology a cure-all or a Pandora's box? Today, we explore one of the most powerful scientific tools humanity has ever discovered, CRISPR-Cas9. This tool can literally rewrite our DNA, and with that power comes both unimaginable hope and serious ethical questions. So what exactly is CRISPR-Cas9? How does it work? What can it do? And should we be excited or concerned? Let's dive in. Let's begin from the basics. Gene editing is a method where scientists can change, delete, or insert specific pieces of DNA, our body's instruction manual. Think of it like using a text editor. If there's a typo in a sentence, you fix it. Similarly, if there's a mutation in your DNA causing disease, gene editing may allow us to fix that. But until recently, editing DNA was extremely complex, slow, and expensive. Then came CRISPR-Cas9, a revolutionary game-changer. CRISPR stands for clustered regularly interspaced short palindromic repeats. Sounds like a tongue twister, right? Let me simplify. CRISPR is a natural system that bacteria use to defend themselves against viruses. Scientists realized this defense system could be repurposed as a powerful tool to edit genes in any organism, including humans. Cas9 is an enzyme, a kind of molecular scissor that cuts DNA at a specific location, guided by a custom-made RNA molecule. You simply tell it where to cut, and it goes there. So, CRISPR-Cas9 equals a GPS plus a scissor for our DNA. Here's how it works, step by step. First, guide RNA is designed to match a specific DNA sequence. This guide RNA brings the Cas9 enzyme to that exact spot in the genome. Then, Cas9 cuts the DNA at that location. The cell naturally tries to repair the DNA. During this repair, scientists can disable a gene like removing a faulty one, insert a new gene like adding a healthy copy, or correct a mutation. Simple, precise, and fast. Let's say someone has sickle cell anemia, caused by a single genetic mutation. Using CRISPR-Cas9, scientists can go to that exact mutation, cut it, and replace it with the correct code. Result: potential cure. The possibilities are truly groundbreaking. Curing genetic diseases, conditions like cystic fibrosis, muscular dystrophy, and even blindness could, be fixed at the DNA level. Cancer treatment is another area, with doctors experimenting on editing immune cells to better attack cancer. In agriculture, crops are being made resistant to disease, drought, and pests, and all that without chemicals. Organ transplants could be transformed too, as CRISPR may help make animal organs compatible with humans. And get this, scientists have even used CRISPR to remove the HIV virus from infected cells in lab tests. This is the future of personalized medicine. Treating disease not just with drugs, but by, honestly, correcting the very blueprint of life. But hold on, just because we can doesn't mean we should. Here's where the ethical concerns begin. Designer babies. Could parents start choosing eye color, height, or intelligence? Inequality is another worry. Will only the rich afford gene editing, creating a genetic elite? And what about unintended consequences? What if editing one gene affects others? Our DNA is deeply interconnected. Germline editing. Changes made in embryos can be passed down for generations. A mistake here could last forever. The real shock came in 2018, when a Chinese scientist claimed to have edited human embryos to make them resistant to HIV, and those babies were born. This raised global outrage and showed how close we are to rewriting human evolution. CRISPR-Cas9 is just the beginning. Base editing is a newer technique that allows changing individual DNA letters without cutting the strand. It's safer and more precise. Prime editing acts like a DNA word processor, kind of like search and replace, but without breaking the DNA. And then there's epigenome editing, which, rather than changing DNA itself, changes how genes are turned on or off. The field is evolving rapidly, and CRISPR is the foundation of a genetic revolution. So could a simple tool rewrite our genetic code and cure diseases we've never been able to touch before? Yes, but it also challenges our ethics, our laws, and what it means to be human. CRISPR-Cas9 is both hope and hazard, a key to curing disease or a doorway to designer genetics. The choice will depend on how we use it, with wisdom, caution, and global cooperation. If you found this video informative and thought-provoking, don't forget, your one like, share and subscribe will help us create more valuable content. Thanks for watching Knowledge Chronicle, where curiosity meets clarity.